Good afternoon. Welcome to your Key NEET English batch. How are you doing? Good afternoon, everyone. Simran, Shauritra, Gnanavi, Bhumika, Sneha, Shravani, Tiso, Tulsi, Madhu, Akshara, Diya, Nikita, Siddhika, Sunandini, and everybody else. Good afternoon. How are you doing? Let me know in the comments if the audio and video is both clear. We have a lot to cover today. Audio and video is both clear. Cool. So in the last class, we had started with the chapter Anatomy of Flowering Plants. We learned about plant tissues. Hopefully you've all studied about it and learnt and remembered everything. So in today's class, we're going to deal with the second part of the chapter. When I introduced this chapter to you, I mentioned that in this chapter, we can split this chapter into two parts. In the first part, we learn about the different kinds of tissues. And in the second part, we will learn about how exactly these tissues are located in the different parts of the plant, like the root, stem and leaves. So in today's class, we will learn about the anatomy of root, stem and leaves in plants in both monocots and dicots. All right. If everybody is ready, give me a thumbs up and we'll get started. Ronika, vanakkam. I think you're feeling that way because I got a haircut done yesterday. It's a fresh cut, brand new. But my husband said I look like a racehorse. Cool. Let's get started. So we are going to be dealing with, excuse me, anatomy of root, stem and leaves in that order of both dicot and monocots. This also is very similar to what we studied in morphology. Okay, so the thing is, it's not difficult to understand. The tricky part is to remember. Okay, understanding is very easy. So we'll finish fast, quickly. Okay. Cool. Okay, pay attention now. Don't get distracted. Pay attention. So first thing, we'll talk about something called the tissue system. Then we learn about the anatomy of different plant parts. I want your 100% attention. Do not get distracted. Okay. So firstly, what is a tissue system? If you think about the levels of organization in animals or in living organisms in general, the most basic is the cellular level. Then you have the tissue level. In plants, we already learnt about how cells will be. We already learnt in our previous class about how tissues will be. Now, eventually, these tissues will come together. Different types of tissues will come together to form organs. In humans or in animals, you have organs like your stomach and your lungs and your heart and your kidneys and brain, all of that. But in plants, what are organs? Organs are your root, stem, leaves, flower, fruit, seed. These are the organs of a plant. So within these organs, there is a specific arrangement of tissues that we call tissue systems. We have three different types of tissue systems: epidermal tissue system, ground tissue system, and vascular tissue system. Now, how do we differentiate these three different tissue systems? First of all, epidermal tissue system. Epidermal tissue system you can compare with the epithelial tissue of us. Okay, if you've done structural organization in animals already, you would have learnt about epithelial tissue system. So not tissue system, in plants, in animals you call it epithelial tissue, right? So what is the function of epithelial tissue? Where exactly is it located? The function of epithelial tissue is to offer protection. And where is it located? It's usually covering the surface or covering or lining the internal organs, right? So basically it's offering protection. It's present on our skin. Epithelial tissue is here, and it's also lining my gut, because if I consume something, then my gut lining has to protect, the lining has to be protected, therefore epithelial tissue is present there. Epithelial tissue is present, lining your trachea as well, it's present lining your alimentary canal as well. So similarly, in plants, this epidermal tissue system function is to protect, okay. So in plants, you do not have organs inside, right? Like we have esophagus and stomach and liver and pancreas and all of that. In plants, you don't find it. So protection is only required on the surface. Internally, lining is not required. But outside surface, just like how our skin protects us, the outer surface of the plants need protection. So this epidermal tissue system is located on the outside and protects the plant. Epidermal tissue system is present in your root, in your stem, in your leaf. It's present everywhere. It is a single layer generally, and it offers protection to the plant. Now we'll talk about the vascular tissue system first, and then we'll talk about the ground tissue system. Vascular tissue system, you can think of it being similar to our, let's say, blood vessels. What do our blood vessels do? The blood vessels and the heart, it's involved in conduction of respiratory gases and also in the conduction of nutrients across different parts of the plant, different parts of our body, right? Very similarly, the vascular tissue system here is composed of xylem and phloem. Remember, xylem and phloem is called vascular bundle, right? So this vascular tissue system consists of xylem and phloem. Xylem helps in conduction of water and minerals. Phloem helps in conduction of food, basically. Now, let's talk about the ground tissue system. Ground tissue system, you can think about it in similar lines with connective tissue. Where is connective tissue present in our body? It's connecting the different organs together. It's making sure that when you jump, your stomach does not come to your lungs, right? So it's keeping everything in place and it's connecting different tissues together. Very similarly, even over here, in between the epidermal tissue system and the vascular tissue system, whatever else is there, that is your ground tissue system. I want you to imagine it like this. You know, in pens, you have a pen's outer body and then you have a refill inside it, right? So think of the refill as the vascular tissue system. Think of the outer body as your epidermal tissue system. Whatever lies in between, that space between the outer body and the refill, over there, it's not free space inside the plant. It is made up of a soft tissue, and that we call ground tissue. Okay, so outer epidermal tissue is offering protection. Innermost vascular tissue is helping in conduction. Ground tissue is everything else apart from epidermal and vascular tissue system. So ground tissue basically fills up all of the spaces within the plant. Okay, is that clear? So these are based on, like I already mentioned, what is it made up of and where is it located. Epidermal tissue system, generally it's made up of parenchyma. Ground tissue system usually has your simple permanent tissues. Such as parenchyma, collenchyma, and sclerenchyma. Vascular tissue system will have your complex permanent tissues. Excuse me. And that is called, that includes xylem and phloem. Majority of the cases, epidermal tissue, it's made up of parenchyma. Remember, I told you that parenchyma is the most abundant type of tissue that you find in plants. Today, we'll learn about why parenchyma is most abundant, where all it's present and all of that. Okay, so now let's move on and talk about each of these systems one by one. We'll start off with the epidermal tissue system. Dermatologist means you know, right? Dermatologist is somebody who is concerned with your skin related issues, right? He's a doctor. You all could be dermatologists, in the future. Derma basically means skin. Epidermal means, epi means above, the outermost layer. dermal layer in the plant that is called epidermal tissue. So basically, as the name itself indicates, epidermal tissue is the outermost covering of all plant parts from the root, stem, leaves, flower, fruit, everywhere epidermis is the outermost layer. Think about outermost layer of anything. It could be the outermost layer of your house. It could be the compound of your house, the compound wall of your house, or it could be the fort wall in a palace, or it could be our skin. What is the function of the outermost thing in any case? In any case, the outermost covering's function is to protect, right? For example, your phones will have a phone cover and a screen guard and all of that. It is protected, right? So even over here in plants, the outermost covering is made up of epidermal tissue and its function is to offer protection to the whichever plant part it is present in. So in this epidermal tissue, what do we find? Obviously in epidermal tissue, epidermal cells will be present. In the outermost layer, what all do we find? Obviously one layer of cells called the epidermal cells will be present. And in certain parts of the plant, in aerial parts of the plant like the leaves and young stems, you can find structures called stomata. You may have learnt about it in lower grades, but today we'll learn about its structure and how it works and all of that. So stomata is present in the epidermal tissue, not all the time. In the roots, it will not be present. It will present only in the aerial parts of the plant. Okay, why it is present only in the aerial parts? When we learn about its structure and function, I will explain to you. Next, epidermal cells, stomata. Thirdly, epidermal appendages. Remember what appendages means? We spoke about appendages in bacterial cell. We spoke about pili and fimbriae and what else, like flagella and all of that, right? So whatever arises from the surface, that is called an appendage. Like for us, our hands and legs, our limbs are our appendages. So it's projecting out from the surface. So such appendages you can find even in plants. If you remember in morphology of flowering plants, we spoke about the different regions in the root of a plant. So in the root, you have a region of maturation from which the root hair will arise. Remember that increases the surface area, water absorption, mineral absorption is enhanced. We studied about all that. So they are basically arising superficially from the outermost layer. Okay, so they are called epidermal appendages. We've learned about epidermal appendages in the root, that is the root hair, and in the stem. If you remember in morphology, I told you sometimes stem and leaves can have hair present on them. Sometimes they are smooth and glabrous. Sometimes you can find hair present, like small thin hair present on the surface of stems. So they are also epidermal appendages. Now there are two things, okay. If you consider a root, if this is like a root will have lateral branches. If it is a taproot system, it will have lateral branches, and also roots will have small unicellular hair, okay. Now these unicellular hair, root hair or hair on the stem, they arise from the epidermis. But these roots or the branches of the stem, they are not appendages. They arise from the deeper part, from within. From within the tissue of the root, they will arise. They are not superficial. They are coming from deep inside. These are superficial. Okay, so these epidermal appendages, we are classifying these appendages as a part of the epidermis because they are arising from the outermost cell layer, which is the epidermis. Whereas that is not the case for these branches of root. Branches of root are different, branches of stem are different. Root hair and stem hair are different. Root hair and stem hair arise from the epidermis. The branches of root does not arise from the epidermis, it will arise from deeper inside the root. Okay, now what is the epidermal tissue made up of? The epidermal tissue is made up of epidermal cells. How are these epidermal cells to look at? See, their function is to offer protection. If your skin is bruised, it is cut in 100 different places, how can it protect you? Effectively, it cannot, because in the air around us, there's so much germs, right? So all of the germs can gain entry into your body and cause infection. If the skin is intact, nowhere it is hurt, nowhere there are cuts, then the skin can do its job well. Likewise, even in the epidermal tissue system, if you have a continuous layer of epidermis, it can prevent germs from entering the plant. Why? What is the issue with germs entering the plant? Similar to how we have infections and diseases, plants also suffer from bacterial, viral, fungal infections and diseases. So it will give an opportunity for that fungus or whatever is the plant pathogen to gain entry into the plant if the epidermis is compromised, if it's broken in certain places. So generally, In the epidermal tissue system, the layer of epidermis that you see, it will have tightly, compactly packed cells present because it has to offer protection really well, right? So over here, you have compactly arranged cells. I want you to notice how there will be parenchyma in multiple places, okay? Depending upon where parenchyma is located, the structure of parenchyma will change. In epidermis, it is compactly arranged. But if you look somewhere else, it will be loosely arranged. If you look somewhere else, it could have certain special thickenings present. So the tissue is of the same kind, but according to where it is present, what is its function, it will show certain modifications. Over here in the epidermis, its job is to offer protection, so it has to be a compact layer without any spaces, right? And obviously, if there is no spaces, if it's compactly arranged, it will be a continuous layer, which means there are no spaces there. No intercellular spaces. Okay, and these are elongated. Elongated means when we draw the section of different plant parts and I show you the epidermis, you will understand mostly it will be like a barrel shape. It will be like this, a continuous thin layer of cells. Excuse me. Okay, so it will look like that. Next, generally it is single layered. Of course, there are exceptions. You won't have to learn about them. But majority of the cases, it will be a single layer. In some conifers, you can see multi-layered epidermis. Okay, I already mentioned this is made up of parenchyma. So how is this type of parenchyma? Parenchyma generally has a thin cell wall, right? Cell wall is not very thick. We already know that. Over here also, there is a thin wall present in these parenchymatous epidermal cells, and there will be a large vacuole in the center. Because the vacuole is occupying such a huge place and you need a nucleus also on the side, the cytoplasm kind of forms a thin layer on the outside. Okay, so there is a small amount of cytoplasm present. Next, now if this is a root, we take a section of the root. How do I show it? Okay, let's say this is the root. Okay, is it visible clearly? It's just like one hollow tube. Okay, so I've taken a section of the root. I'm focusing only on the epidermal layer. This is the outermost layer, which is the epidermis. Forget about what lies inside. We're not yet talking about it. Okay, so this is the outermost layer. If I draw this outermost layer here like this, epidermis, this is your epidermis. This is one layer of cells. This epidermis will have an inner side and it will have an outer surface, right? If cells are lining this, you will have inner surface of cells and you will have outer surface of cells. You will find cuticle, you will find epidermal hair, root hair or stem hair arising from the outer surface. When we say on top of epidermis there is a cuticle, we don't mean it's present on the inside. Again, why is it not present inside? Because the function of the cuticle is to reduce the rate of transpiration. By being present on the inside, it will not make any difference, right? If it's present on the outside, it will become like a water impervious layer. It will become a waxy layer and it can reduce the loss of water. If it's present on the inside, what does it do? What does it help in? It's a waste, right? So over here on the outside surface, you can find two things. You can find the cuticle and you can also find root hair or stem hair. So they are present only on the outer surface. A thin layer of cuticle would only be present on the outer surface over here. Epidermal hair, either root hair or stem hair, would only arise towards the outer surface. If it's present on the inside, does it make any sense at all? It doesn't, because absorption happens from the outside. Its requirement is only on the outside. So it need not be present on the inside. Okay. So yeah, outside surface, you will have a cuticle Cuticle, I'm sure you've learned in your lower grades, cuticle is a waxy substance. It's a fatty, waxy substance. So this is like Vaseline. We apply Vaseline during winters because you do not want water loss. You do not want dehydration of your skin, right? So that similarly, even the plants have an inbuilt Vaseline. They don't need anybody to apply Vaseline on them. They have a cuticle already, and that cuticle will help in reducing water loss. It will help in reducing the rate of transpiration. Basically, it will not allow the plant to go dehydrated. Okay, so that prevents water loss. Okay, I told you that cuticle's function is to prevent, not necessarily prevent, reduce the rate of transpiration. It reduces water loss. Water loss happens wherever it is exposed to the outside. In roots, stomata are not present, cuticle is not present, because over there water loss by transpiration will not take place. Okay, so remember this point, cuticle is absent in roots. Do you remember what we learnt in the last class? Like I told you that vessels are not present in gymnosperms, sieve tube elements in companion cells are not present in gymnosperm. Some things that are absent in monocots, some things that are absent in primary phloem. Do you remember all of that? Those are important. So in that list, you add this also. Cuticle is not seen in roots of plants. Okay. So yeah, we've spoken about how the epidermal cells are. What is the cuticle? Where is it present? What is its role? Now let's talk about root hair and stem hair. These are your epidermal appendages. We already know about root hair. They are very small. One important thing about root hair is that they are unicellular. The difference between the hair that you find on roots and the hair that you find on stem is, in stems it is multicellular, in roots it is unicellular. What does that mean? If this is one epidermal cell in the root, one cell will give rise to one root hair. This is root hair. In stems, you will have the epidermal cell, and this epidermal cell will give rise to stem hair. But this stem hair is not a part of this cell. This stem hair within it will have more than one cell present. So this is a multicellular structure, and we call it trichomes. Trichomes are nothing but your stem hair, or whatever you find on leaves, that also can be called trichomes. The difference between root hair and stem hair, function is different, number of cells present in it is also different. In roots, one cell, from the cell itself as an extension it will arise. In stem, it's a multicellular structure. In root, what is the function of root hair? It increases the surface area available for absorption of water and minerals. It enhances the rate of absorption basically. Whereas in stem, these trichomes basically help in reducing the rate of transpiration, because the stem need not absorb anything. If there is no absorption happening in the stem, what is the function of these trichomes? Because of the presence of these trichomes, transpiration rate is reduced. Okay. Transpiration again is the loss of water. Cuticle and trichomes have the same function. Both of them work to, save more water and lose less water. Okay. So yeah, root hairs are unicellular elongations. They help in absorption of water and minerals from the soil. Stem hair are called trichomes. Generally, they're multicellular. I think they're always multicellular. Yeah, they may be branched or unbranched. Sometimes these hair can have branches, like split ends we have, no? Like they can be branched or unbranched. In some plants, you can see that it's soft. You won't even notice it's like very soft kind of furry kind of thing. But some plants, it's very stiff also. Sometimes they can be secretory as well. They will secrete certain substances. Basically, its function is to prevent or reduce the rate of water loss because of transpiration. Okay, so that's all about the root hair. Now, the most important part about the epidermal tissue system is the stomata. Stomata, we know, are pores that are present on the surface of the aerial parts of the plant. Stomata can be found in very young stems on the surface. Because it's on the surface, because it's in the epidermal layer, we consider that under the epidermal tissue system. And in leaf, of course, stomata has to be present because that's how exchange of gases will take place. Now, these stomata you may have learnt as pores that are present on the surface of the stem and young stems and leaves, but these pores are not forever open, okay? Depending upon the water status of the plant, depending upon how much water it is getting, is the conditions in the environment very favorable for transpiration, is the plant losing more water than it should, depending upon the conditions in the environment, the plant will decide whether the stomata has to be kept open or if it has to be closed, okay? So these pores are not open all the time. There are specialized cells that guard the opening and closing of stomata. Okay, so the stomata, when the conditions are not favorable for the plant, it will close shut. And when the conditions are good, when there is not much transpiration, the plant is getting sufficient amount of water for photosynthesis, then it will stay open. Depending upon the environmental conditions, depending upon the time of the day, stomata will open and close. It's not forever open. All right. So that's one thing. Now, the stomata, like I told you, stomata basically, the moment you listen to the word stomata or hear the word stomata, you associate it with a pore, right? So the pore or the stomata is called the stomatal aperture, or you can call it the stomatal aperture. Excuse me. Okay, I told you that this stomatal aperture, this is the stomatal aperture, and here you have the stomatal aperture. How many times I'm saying stomatal aperture? Okay, so here you have the stomatal aperture. Surrounding the stomatal aperture, you have cells that will regulate the opening and closing. So these cells are like security guards in your apartment building or in your office or in your college. What does the security guard do? Security guard makes sure it will let in the right people inside. Unless you show your ID card, you will not be allowed inside your college or school, right? And random people cannot enter the institution if it's a college or school. And then you can't leave midway while classes are still on. He will not let you go outside the college gates. So a security guard's job is to ensure when to open the gate, whom to open the gate to, and all of that. So these cells here do a similar job. So we call them guard cells. Guard cells always occur in pairs. It's one jodi. Okay. So it's like Shah Rukh Khan and Kajol. So it's like one pair all the time. Guard cells always occur in pairs. Now the shape of these guard cells is different in monocots and dicots. In dicots, they are kidney shaped or bean shaped, you can call them. And in monocots, they are like your dumbbells. Okay, so it is dumbbell shaped. These are your two guard cells. I will outline them in dark green color. Okay, so this is one guard cell. This is the other guard cell. You can see how it is bean shaped or kidney shaped. This is in dicots. And over here, they are in dumbbell shape. Right? So this is one, this is two, this is one, this is two. That's always a pair. In monocots, dumbbell shaped. In dicots, it is kidney shaped or bean shaped. All right. Now, these are called guard cells. Now you're looking at it from above. For example, this image that you're looking at the surface of a leaf from above. So from the surface, you can see there are pores present. But if you look from the side, it will look like this, a continuous layer of epidermal tissue. Okay, so it's a continuous layer like this, right? This is from the side you're looking at it. You will see some cells that the shape of those cells will be slightly different from these cells, okay? And that will form a pore. See, if this is your leaf. Over here, in this diagram, you're looking at it like this. But over here, I'm showing it to you like this. Remember, epidermis is present here and here. So on top layer, I'm showing you, this is the epidermis. Many cells are present. In between, there are specialized cells. The shape of this cell is different from the shape of the other epidermal cells. They are your guard cells. Okay, so you take this and look at it like this. Here's what it will look like. Okay, now let's talk about the guard cells. The guard cells, like all plant cells, will have a cell wall, and the cell wall is differentially thickened. That means its thickness is not the same throughout. In certain regions, it's more thick. In certain regions, it's more thin. Okay, so here it is differentially thickened. Where is it more thick? Where is it more thin? Let's see that. Towards the stomatal pore, over here, the cell wall is thicker. Comparatively, the outer wall is thinner and more elastic. Same thing even there. Now, surrounding these guard cells, I want you to notice the other cells that are present. Pay attention. This is also an epidermal cell. This is also an epidermal cell. This is also an epidermal cell. Most of your epidermal cells look like this. Like this. Okay. Guard cells are specialized. They look like this. Now, the cells of the epidermis in the immediate vicinity of guard cells, right next to the guard cells, like the immediate neighbors, not like 2 streets away, not like 2 houses away, just the house next to yours, like immediately next to the guard cells, you will find that some epidermal cells look a little different, right? They are specialized and they will appear a little different from the other epidermal cells. These have a special name. We call them subsidiary cells. Now, how or when does the stomata open and close? Whenever, let's say, the water status in the soil is good, there is sufficient amount of water, even in dry days, even on very sunny days, the plant is getting sufficient amount of water, there's no water stress. So what happens is, from the epidermal cells, the subsidiary cells, the surrounding subsidiary cells, water will enter into the stomata. Into the guard cells. The guard cells will take in all of that water, endosmosis happens, and it will start swelling up. I told you that this outer layer of outer wall is elastic, it is thin and elastic. So when more and more water enters, it will start expanding. As it expands, it will pull these thick walls away and create the pore. What happens when there is little water, when it's too dry, when the temperature is very high, the plant is not getting sufficient water, it wants to conserve water and not lose it in transpiration. What it does is the exact opposite. When the temperature is very high, when the conditions are not favorable, this water from the guard cells will be lost into the subsidiary cells. Exosmosis happens in the guard cell, it will lose water to its surrounding cells, and then this will become flaccid again. When it becomes flaccid, the stomatal pore closes shut. When it is turgid, the pore will open. When it's flaccid, it will close shut. Okay, so yeah, that's about how the stomata works. Another important difference that you should remember about a stomata and other epidermal cells is, other epidermal cells will not have chloroplasts, they will not perform the function of photosynthesis. The guard cells do have chloroplasts, they can photosynthesize, the other epidermal cells will not have it. Okay, so yeah, that's about the epidermal tissue overall. Stomatal aperture. This stomatal aperture plus the guard cells plus your subsidiary cells together constitutes what is called a stomatal apparatus. Okay. The same thing I've given in text here. Stomata are structures present in the epidermis, regulate the process of transpiration. It is composed of either 2 bean-shaped or dumbbell-shaped cells. Outer walls are thin, inner walls are very thick. Guard cells have chloroplasts. Guard cells basically regulate when the stomatal pore should open and when it should close. So if you have any doubts, let me know. Kanak Malik is asking, you didn't get the point. Trichomes prevent water loss due to transpiration. Transpiration is basically when water that is present inside the plant is lost in the form of vapor. That is called transpiration. Water is important for the plant, right? For photosynthesis, water is essential. But how does water from the soil reach the leaves against gravity? To pull water up to the leaves, energy is not utilized, but transpiration pull is utilized as a force, right? So transpiration is important for the plant. But if all the water that the plant is absorbing is getting lost, we do not get sufficient water for photosynthesis at all. The plant will not get food to eat, right? So transpiration has to be balanced. Transpiration should be just enough to pull enough water to the leaves. So sometimes when the humidity is very low, when the temperature is very high, when there's not sufficient water in the soil, more water will be lost as transpiration and the plant will not be getting sufficient water for photosynthesis. In such cases, plants will have certain modifications. One of the modifications is that, or adaptations you can call it. One such adaptation is having a very thick cuticle so that water cannot escape, it will form a waxy layer, or having trichomes or stem hair or hair on the leaf that will prevent the rate of transpiration and help save water for the plant. So yeah, that's it. Yes, it is trichome only, Diya. Trichomes are present on the stem and on the leaves also, Janani. Akshata did not understand closing and opening. Okay, I'll explain. See, think of guard cells like a balloon. Okay, so if you add more water into it, it will swell up, right? And if the balloon loses water, it will become kind of flaccid. Same way, even in the stomata, the guard cells, it may lose water or it may get water. If it's getting water, it will get it from the subsidiary cells. If it's losing water, it will go into the subsidiary cells. If water is entering into the guard cells, it will expand. It will become bigger and it will stretch out. When it stretches out, that pore will open. When the guard cells will lose water into the surrounding subsidiary cells, it will become flaccid and the pore will close shut. Hopefully tomorrow it will be done, Srijana. The shapes should be in such a way that a pore is created in the center, right, Kavina? Think about it. If they are two spherical cells, a pore will not be created in the center. It should be in such a way that it can create a pore in the center. Dumbbell-shaped cells can create a pore, and bean-shaped cells can create a pore. The reason why we are the way we are, or any plant is the way it is because over the course of evolution, it would have tried and tested different kinds of things. Whatever works for it best, it would have stuck to it. And that is what continues. When you learn about the chapter evolution, you will understand why anything is the way it is. It's not like the plants would not have experimented with the different shapes for guard cells. Of course, in a population of plants, when guard cells developed, probably some of them may have been spherical, some of them may have been rectangular, some of them may have been like the dumbbell-shaped cells or your bean-shaped cells. You know, in bean-shaped cells, it works best. So that population survived better than the rectangular cells or spherical cells. So those genes were transmitted to the next generation because they survived better, and that got continued, right? It's basically whatever works for it best will stick on. More like natural selection, you can say. Yeah, survival of the fittest, yeah. None that I have seen. Cuticle may be present on fruits and vegetables, but I've trichomes, if you're eating the stem of some plant, then probably trichomes will be present in the stem. I don't know. Vegetable stems can be vegetables, right? Like potato is a vegetable, it's a stem. Cool. Let's move on then, if everything is clear. First we'll study about vascular tissue system, then we'll study about ground tissue system. Okay, but in your textbook it's first ground and then vascular. Okay, but we'll do vascular first. Okay, so the vascular tissue system basically consists of your vascular bundles, which is your xylem and phloem. What is its function? Its function is to help in transport of water and minerals and food across different parts of the plant, right? So vascular bundles, depending upon how they are arranged, you have different kinds. One type is radial and the other type is called conjoint. In radial type of vascular bundle, xylem and phloem are present in separate bundles. In conjoint, they are both present together, joined together. Radial, separate. In radial vascular bundle, this is how it will be. How did we get to this point? Think of a pen as the root. You're taking a section of the pen. I told you that the outer body is the epidermis. The inner refill is like the vascular tissue. That is where the vascular bundles are present. So what you're looking at here is the inner refill. When we learn about actual root, stem, leaf section, then you'll understand where this image is coming from. So you can see over here how xylem is present here, 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 here. And you have phloem present here, 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 here. They are not present together in the same bundle. Why are we calling it radial? You know how the radius is for a circle, right? From the center to the circumference, that is your radius. If you look at this radius, you have xylem. This radius, you have phloem. This radius, you have xylem, you have phloem. It's alternating, but it's not present in the same radius. Draw a radius anywhere and you do not find both xylem and phloem there. Either you find xylem or you find phloem. And xylem and phloem will always alternate with each other in radial vascular bundles. Where can you find radial vascular bundles? You can find them in roots, both dicot and monocot. Okay. Now let's talk about conjoint. Conjoint will have both xylem and phloem in the same bundle, in the same radius. So if you take this as a central portion of a stem, let's say for example a dicot stem. Okay, let's see, this is a dicot stem. In a dicot stem, this is just one vascular bundle. Okay, so these are one vascular bundle. Oh God, what is wrong? Vascular bundle. Within the stem, many such vascular bundles will be present. For example, in a dicot stem, vascular bundles will be arranged like this in the form of a ring. Okay, like this in the form of a ring, it will be arranged. Over here, you can see that towards the inside, you will have xylem. Towards the outside, you will have phloem. Have xylem, phloem. Xylem inside, phloem outside. Xylem inside, phloem outside. Xylem, phloem, and so on. Now look at the radius here. Take any radius. Look at this, you have xylem and phloem both in the same radius. Look at this, you have xylem and phloem both in the same radius. Both in the same radius. So when you're having it both in the same radius, they're present in the same bundle, we call it conjoint. Now within conjoint, you have two types, closed and open. What does closed means? What does open mean? Closed vascular bundles are conjoint. We call them conjoint closed. In conjoint closed, within phloem and xylem, you do not find cambium. Remember what cambium is? Cambium is a type of meristematic tissue that is present laterally. I told you it's present only in dicots, right? So these closed vascular bundles, cambium is absent. You find them in monocot stems. In open vascular bundles, you have phloem facing the center, xylem facing the center, phloem facing the outside, and in between the xylem and phloem, why did I write? And in between the xylem and phloem, you will find a strip of meristematic tissue which is called cambium. Cambium is present. Okay, so this can be seen in dicot stem. So what does the presence of cambium have to do with it being called open and closed? Why is it closed if there is no cambium? What is it open to if cambium is present? We need to understand this. Now what happens is, if you take a dicot stem, okay, I am showing you only the vascular bundles. You have vascular bundles arranged in the form of a ring like this. Okay, so on the inside, you always have xylem, 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 xylem. On the outside, you always have, what color should we use? I haven't used green in a while. Phloem, 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 phloem. In between the xylem and phloem, you have a strip of meristematic tissue which is called the cambium. Okay, this is your cambium. Cambium is a type of lateral meristem. Okay, so this cambium, if it's meristematic, obviously the cells in this cambium will have the ability to undergo cell division. Remember how we classified tissues? The ones that can undergo cell division are called meristematic tissues. The ones that lack the ability to undergo cell division, they are called permanent tissues, right? So you have a strip of cambium present here. Now these cambial tissue is present between the vascular bundles. It's present within the vascular bundle basically. So we call it intrafascicular cambium. It's present within the vascular bundle. Intra means within. It's present within the vascular bundle. So it's called intrafascicular cambium. Now in between this cambium, over here, between the, vascular bundles, not between the cambium, in between the vascular bundles, you will have parenchymatous tissue. Okay, so this is already differentiated, that cannot undergo cell division. But in the same line as our cambium, some parenchymatous tissue here will regain the ability to undergo cell division and become meristematic again. Just these. Okay, otherwise, apart from these orange cells above and below, it will be parenchymatous only. But just in this line, parenchymatous cells will regain the ability to divide. It will get de-differentiated and become meristematic again. Now that is called interfascicular cambium. Remember when we spoke about lateral meristem, you learnt few examples of intrafascicular cambium, interfascicular cambium, cork cambium and all of that, right? So this is intrafascicular cambium that is there from the beginning. Interfascicular cambium, by a process of de-differentiation, it will form. Now both of these will have the ability to divide, and they will start dividing in this angle. Alright, so as they start dividing in this angle, more cell layers will get added, right? Cell will get added on the inside, cells will start forming on the outside as well. Okay, so when these meristematic cells, cambial cells, will start undergoing cell division, it will add more cells to the inside as well as the outside. What will happen as a result of that? This stem will increase in girth. It will become wider because of more cell layers getting added to it, right? So that kind of growth is called secondary growth. Secondary growth happens only in dicots because of the presence of cambium. Secondary growth basically is increase in girth. Increase in girth. Think of a dicot plant or a tree like a mango. You sow a mango seed, it will become a small sapling. It has a very thin stem. But as this mango sapling grows, of course, it's increasing in height. It's becoming taller and it's also becoming wider. The stem is becoming wider. The reason why it's becoming taller is because of the meristems that are present at the apical ends. It is present at the root tip and the shoot tip, that is contributing to its increase in length. But if you think about why it's increasing in girth, it's increasing in girth because of this cambium that is adding more layers inside and outside. Think of a monocot plant. In a monocot plant, cambium is not present. Think of a bamboo plant. Does the bamboo stem become like a banyan tree trunk or a mango tree trunk? It doesn't. Does a sugar cane become like a mango tree trunk or wheat or maize or any plant you take, monocot plant, it will not grow in girth. Secondary growth will not happen. Why does secondary growth not happen there? Because there is no cambium. Because there is no cambium, they are closed to secondary growth. Because of the presence of cambium, dicots are open to secondary growth. Okay, so that's the reason why we call them open and closed. Okay, when we say it is conjoint open, that means secondary growth can happen there because of the presence of cambium between xylem and phloem. When we say it's conjoint but closed, we mean they're present in the same bundle but there's no cambium, so they cannot undergo secondary growth. So secondary growth is basically increase in girth. This will happen only in dicots. Primary growth this will occur in monocots as well as dicots. This is because of apical plus intercalary meristems, and this is increase in height or length. Alright, so that's the reason why we call it closed, because you're close to secondary growth, open because you're open to secondary growth. Alright, now let's learn about something else in xylem. I told you that in xylem you have protoxylem and metaxylem. If you take vascular bundles, the diameter of the xylem elements will vary. Right, whichever is first formed will have a smaller lumen, whichever is later formed will have a bigger lumen. This is the center of the plant, whichever part, and this is the peripheral region. The one that has the smaller lumen is called protoxylem. The one that has wider lumen is called metaxylem. Protoxylem is first formed, small lumen. Metaxylem is later formed, it will have a larger lumen. Now, depending upon position of protoxylem, depending upon the position of protoxylem, you can categorize this into two types. If protoxylem, excuse me, if protoxylem is facing the inside, as you can see here, protoxylem is inside, metaxylem is outside. If protoxylem is inside and metaxylem is outside, such a type of xylem is called endark. Endark. Endo means inside. What are we classifying this based on? Based on protoxylem. Protoxylem here is facing the inside, so it is endark. Here, protoxylem is outside, metaxylem is inside. Since protoxylem is outside, we call it exarch. Exo means what? Not dishwash bar. Exo means outside. So this endark kind of xylem we generally find in stem. Exarch you find in roots. Okay? Endark and exarch is only specific to xylem. Open and closed radial, that is with respect to both xylem and phloem. Endark exarch is only about xylem. Alright? That's about your vascular bundles. Now, apart from epidermal tissue, vascular tissue, whatever else is present in the plant, that is your ground tissue system. So, ground tissue system, like I already mentioned earlier, consists of simple permanent tissues like parenchyma, collenchyma, sclerenchyma. Where all are they present? They are present in the cortex, pericycle, pith, medullary rays. You don't know any of these terms. So, we learn about it later as we go on. Right now, don't worry about what these mean. In leaves, the ground tissue is called, it has a special name, we call it mesophyll. When we learn about leaves, I'll tell you what this is. So that's all about your epidermal tissue system. If you have any doubts, let me know. Okay, I will move on. No doubts, no? Yes, annual rings are formed due to secondary growth. Open conjoint, yeah, gymnosperms also show secondary growth, so it's present. Yes, Taufik, it is there. In radial also it can happen, but only in dicots. Tissot. Girth means increasing in diameter. It will become wider. Ajay, any cambium will not develop in monocots. Intra or inter, both are absent. Secondary growth I will not teach because it's not part of your new syllabus. It's a wonderful concept, but unfortunately they've deleted it. Alright, let's move on. Okay, now let's start off with the root. Today we'll learn about the root. I think that's all we can learn today. If possible about the stem, or enough, we'll do just root today. Tomorrow we'll do stem and leaf and PYQs of this chapter, and if time permits, morphology of flowering plants as well. Okay, so yeah. This is a transverse section of a dicot root. First we learn about dicot root, then we learn about monocot root. What in dicot root should we learn about is the section, what kind of tissue is located where, what is its function, and all of that. Now, how did we get to this diagram? Let's understand that. This is a root. Let's say this is a root. You're taking a very thin slice of a root, like you cut carrots. You cut roots, but very, very thin slices, and you place it under the microscope. You will get a structure like this, right? You will have an outer layer, which is the epidermis. Inside, you will find vascular bundles, right? So here is the epidermis. Here is where you will find vascular bundles. In between that is your ground tissue. Now, I don't have to draw this entire thing to explain to you how tissues are arranged. If I just take this one pizza section like this, that is how I will get this. Okay, so whatever I'm telling you is present here, the same thing will be present here also. Whatever is the tissue that is present here, the same thing will be present here as well. Whatever I'm showing in the vascular bundles, I'm showing entirely. So that's okay. So we get this diagram. From this diagram, you're taking one section or one portion of the root section and we're studying this. The same thing will apply throughout. Okay. So any part of the plant, any part of the cell that we're studying, remember how did we study about cells? We studied about cells from the cell wall, then cell membrane, right? And then you study about organelles. How do you study about organelles? Outer membrane of mitochondria, inner membrane of mitochondria, what's present inside it. So whenever you're studying the structure of something, most cases we start from the outside to inside. Even here, we will learn from the epidermis and then we will move on into the ground tissue and then about the vascular bundles and all of that. All right. So yeah, this is a dicotyledonous root. Outermost layer is your epidermis, right? Outermost layer is the epidermis. This is an actual section of your NCERT textbook. This is your outermost layer. You can see how cells are arranged very compactly. They are barrel shaped like I showed you. And from some cells, there are extensions or appendages arising. They are nothing but your root hair. In the root, do we have cuticle in the epidermis? No. In the root, do we have stomata? No. So what do we have? We have continuous layer of epidermal cells and here and there you find some root hair. The epidermis of roots in both monocots and dicots has a special name. We call it the epiblema. Epiblema is another name for epidermis that is only for root. Epiblema you can't say for stem or leaf. The root epidermis is also called epiblema. So over here many cells protrude in the form of unicellular root hair. I've already told you about that. Now what lies inside is the ground tissue. Here we call it the cortex. The cortex also is made up of parenchyma. Now I want you to understand how even in the epidermis it is parenchyma, even in the cortex it is parenchyma. But look at how different the two cells are, the two tissues are. Despite being parenchymatous, they show difference in the structure. Over here it is barrel shaped in a continuous layer. Over here it is almost spherical or isodiametric with a lot of intercellular spaces, right? So yeah, also in the epidermis it is a single layer. In cortex you can see many layers present. So from here to here it is cortex. This also is cortex. So you have many cells present, multiple layers of cells present. They are almost spherical. They have thin walls and you can see there are a lot of spaces that are present between cells. They are called your intercellular spaces. They are thin-walled cells with intercellular spaces. Why should intercellular space be present here? Remember, every living cell has to undergo cellular respiration. For cellular respiration to take place, oxygen is required. Where is the plant getting oxygen from? It will get it from some pores that are present on the outer surface. So through that, oxygen will enter. But if everything is compact here, how will oxygen reach the cells? For oxygen to reach the cells, carbon dioxide to escape the cells, spaces are required, right? So that is why this will give the cortical cells will have a lot of intercellular spaces. When we learn about photosynthesis or even respiration, not photosynthesis, when we learn about respiration, I will explain to you why this is important. I will ask you, then you should tell me. Okay, so that's about your cortex. Next, we have, wait, I think I forgot one thing. I didn't talk about our endodermis at all. Cool. So, the innermost layer of the cortex that is there, innermost layer of the cortex, it is called the endodermis. Epi is above, outer, excuse me, epidermis, and inside, innermost layer of the cortex, that is called endodermis. Endo means inside. Similar to how epidermis is a continuous layer on the outside, within the root, you will have a continuous layer on the inside which is called the endodermis. Endodermis in roots is very, very important. I'll tell you why. Okay, so this also similar to epidermis is a single layer. This also similar to epidermis has barrel-shaped cells that are made up of parenchyma. Similar to epidermis, even here there are no intercellular spaces. But what is different here is in these endodermal cells of the root, you have Casparian strips present. Now, different cells will have cell walls a little different, right? For example, if you consider parenchyma, obviously it will have excuse me, cellulosic cell wall, and hemicellulose may be present. But if you take Collenchyma, pectin will be present. If you take Sclerenchyma, in the secondary wall, lignin will be present. So here parenchyma is slightly modified. Parenchyma of course has a cellulosic cell wall, but along with that it will have another material, another substance that is called suberin, suberin, that is also a polysaccharide. It will have suberin. Now this suberin is not present in all walls of the cell. A cell wall, let's say, I don't have anything to show it. Let me take this. Okay, let's assume this is a cell. Okay, so this is one cell present in the endodermis. If this is one cell, let me draw it here. If this, let's say this is one cell. This cell will have 6 walls, right? If it's a 3D structure, it will have 6 walls. 1, 2, 3, 4, 5 and 6. It will have 6 walls. So let's assume this is the cell. In the radial walls of the cell, radial walls means draw a radius from here. If you're drawing a radius from here, which wall will be along the radius? Those walls are called the radial walls. If this is the cell, this wall and this wall, these are called the radial walls. Okay, so in the radial walls of these endodermal cells and also in the tangential walls, how do you draw a tangent to a circle? You draw a tangent like this, right? These are tangents. So if you take this cell, this is the radial wall, this wall and this wall is the radial wall, this wall and this wall, this and this, these are tangential walls. Only in the tangential wall and the radial wall, these 4 walls, you can find the deposition of suberin. You will not find it here, you will not find it here. In these lateral walls, you will not find. So here, 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 in these radial and tangential walls, you have suberin deposited, and that is called Casparian strips. You were learning about what is the importance of Casparian strips in a chapter called Transport in Plants, which was earlier there, but right now it is deleted, so let's not worry about it. Basically, the suberin is impervious to water. Okay, that's already written here. Water impermeable. Like cuticle, this is also a waxy material. It will not allow water to pass through it. Like if it's present here, water is getting absorbed from here, right? It will get absorbed, it will come here, it will come here. But here there is a thick wall over here. It cannot easily pass through this. Okay, there are special cells called passage cells which will not have it. Through it, it will pass. Basically, endodermis acts like a checkpoint. Water can carry along with it a lot of things, right? So in the endodermis, it gets checked what should be allowed to enter the vascular bundles, what should not be allowed to enter into the vascular bundles. Because once it's entered the vascular bundle, it will go throughout the plant. So this basically acts like a checkpoint. I want you to remember about endodermis and roots, both monocot and dicot, you have suberin deposition in the cell walls. In the radial and tangential walls, this suberin is a waxy material that is impervious to water. Alright, so that is the important thing about roots endodermis. And this endodermis is the innermost layer of the cortex. Now ultimately talking about vascular bundles, here you have xylem, and outside, in between, you have phloem. Okay, this is xylem and this is phloem. What kind of arrangement is this? If xylem and phloem are present in alternate radii, they are not present in the same bundle, what do we call it? We call it radial vascular bundle. So here you can find radial vascular bundles. The number in dicot varies from 2 to 4. It can be 2 least or 4. Look at the xylem now. How is the xylem arranged? Where is the protoxylem facing? Protoxylem is the smallest vessel with the smallest diameter. Is it facing outside or is it facing inside? It's facing outside. If it's facing outside, what is it? Exarch condition. So xylem is exarch. Okay, that's about the dicot root. Dicot roots, vascular bundles. Now, in between the vascular bundles, if this is phloem, 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 and over here you have xylem, 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 xylem. In between that, over here, you will have parenchymatous tissue that is called conjunctive tissue. That is present between xylem and phloem, that is called conjunctive tissue. In the center, whatever is present, that is called the pith. This is the pith. The pith is very small here, inconspicuous, you can't see it very clearly. Now over here, epidermis is parenchyma, cortex is parenchyma, innermost layer of the cortex, endodermis is parenchyma. What is that? Conjunctive tissue is parenchyma, pith is parenchyma. The only thing that is not parenchyma is your vascular bundles. Okay? Wait, why did I not talk about the pericycle? Where are you, pericycle? Oh God. Okay. So this is the epidermis, and then you have the cortex. Innermost layer of the cortex is the endodermis. Inside the endodermis, you have a layer that is called the pericycle. Peri means around. Around this circle, vascular bundle, whatever is present, that is your pericycle. Should I draw it in? Okay. So over here, inside the endodermis, you will have a layer of cells. That is your pericycle. Now, pericycle also has certain important points like endodermis and root. You have to learn about it. It's very important. Initially, in today's class itself, I told you that from the root hair also arise. From the root, branches also will arise. I told you that the roots arise superficially from the outermost layer. Branches arise, secondary branches of the root, all of that will arise from deeper. So that will get initiated over here. So it will start arising from the pericycle. Okay, like how epidermal hair arises from the surface superficially, but this will arise more deeply. So initiation of secondary roots or branches of roots will happen from the pericycle. That is one important point. And in dicots, there is increase in girth of the stem because of secondary growth. Same secondary growth will happen even in roots as well. But the roots don't already have cambium. Cambium has to be formed. Similar to how interfascicular cambium formed from parenchyma, over here, interfascicular cambium partly will form from pericycle. Okay, so yeah, initiation of vascular cambium will happen in the pericycle itself. Alright, so if you have to talk about endodermis, it's Casparian strips in the root. If you have to talk about initiation of lateral roots and initiation of cambium, vascular cambium during secondary growth in the root, that will happen because of pericycle itself. That's about your dicot root. Now let's talk about monocot root. There is only two differences between monocot root and dicot root. First difference is with respect to the number of vascular bundles. In dicots, we just saw it can range from 2 to 4. But in monocots, it's more than 6. Okay, so there will be more than 6. At least 6 will be present, or more than 6 will be present. That is called polyarch. Poly means what? Many bundles are present. Over here, another difference is the pith is more conspicuous. There you cannot see it very clearly. Actually, this diagram is not very good. In monocot root, you can see a bigger pith, but here they've shown it in the same size. But in the text, they've given that pith is inconspicuous in dicot and conspicuous in monocot, but the diagram is not doing justice to it. So that is the only two differences you have here. In dicot roots, in monocot roots, you have fibrous root system. So over here, the pericycle will not give rise to lateral branches because the roots will arise from the base of the stem. Over here, secondary growth will not take place. So the pericycle will not give rise to the vascular cambium as well. Over here in endodermis, Casparian strips will be present similar to dicot root. That is the only difference. Alright, so I think that's it. We should stop at this point. Let me know if you have doubts. Kirtana, pericycle and pitta are also in the region of xylem and phloem, but they are ground tissue. Yeah, pith and pericycle are ground tissue. Everything other than epidermis and xylem phloem is ground tissue. Cortex is ground tissue, endodermis is ground tissue, pericycle is ground tissue, conjunctive tissue is ground tissue, pith is ground tissue. Apart from xylem and phloem, everything else is ground tissue. Questions we'll do tomorrow, Gunjan. Simran, heart and anchor. Thank you. I've spoken so much today, I feel like I would have burnt 1000 calories. Tawfiq, the topper in the polls, he's asking for more questions. We'll do questions tomorrow. Okay. Kirtana, pericycle and cambium. Cool. See, what happens in dicot root is they don't already have cambium. Cambium should form. So cambium will form from pericycle, right? So some cells of the pericycle will form a cambium. A cambium basically forms a wavy ring like this. Well, okay. So it will form a wavy ring like this. Okay. So over here, whatever pericycle cells are there, they will undergo dedifferentiation and become meristematic and form cambium. So those cells will divide and give rise to increase in girth of the root. That is what I meant. Pith in monocot sakshata is slightly bigger compared to dicots. That is what they've given in the text. But in the diagram, they've shown both to be of the same size. But if you take a section of a monocot root and a dicot root and look under the microscope, you can clearly see the pith is bigger in monocots compared to dicots. That was in the stem kirtana. This is in the root. In the root, it can't be present between xylem and phloem because xylem and phloem themselves are not present together, right? So this is root. Pratik, I'm looking forward to meeting you all too. Hey, how did your tests go on yesterday? How did you all do? Has anybody got full marks in botany? Let me know. That'll make my day. My favorite biologist. Actually, I'll tell you who is my favorite biologist. You know, in BBC Earth, there is this one person called Sir David Attenborough. I like him a lot. He's my most favorite person. Yeah. I got interest in studying plants and animals because of what used to play on BBC Earth. My dad, every time he used to play that on TV, and every time something interesting comes in, he would call me, come sit and watch this, come let's sit watch this. So I used to see that a lot and I like nature a lot. Aha, very nice. Divya's got full marks. Congratulations. Shahid, you can't say only one question. One question also matters. You barely had like 12-15 questions for the test, right? And it was only one chapter. I know it had a lot of examples, so I made sure I'll give you only one chapter. Okay, Shreya also has got full marks. Congratulations. Divya has by hearted everything. Yes, Prasanna, I have visited Tirupati, Tirumala, everything. My favorite hero is Dr. Rajkumar, Kannada hero. He's very old. He's like in Hindi, you have all of these Raj Kapoor and all of them, no? Like that, from that time, he is my favorite hero. Enough now. Taking my interview. We'll finish today, okay? Does anybody have any doubts in what was taught today? Vagdevi, congratulations, you've also got full marks. Mehak has got full marks. Wow, Ramya has got full marks in all the subjects. I think we should all just clap for her and congratulate. Sahiti, even 1 mark, one wrong question can cost you a lot. That is what I mean. Okay. All right then. I'll see you in tomorrow's class, sharp at 12 o'clock. Okay? Bye-bye. Take care. Can you please tell on which portion you should lay stress in this chapter, Pratik? Every line, every word of NCERT, you should lay stress. Oh, Tissot also has got full marks. Perfect. Very good. Congratulations. Where is my heart of hearts? Damn. Tomorrow we'll finish this chapter, but please study whatever was taught today. Read it today itself. Also, it's like 2.5 months since we started this batch. I finished like pretty much 35-40% of the botany syllabus. I mean, 11th grade. No, put together, let's say 30%, 35%. How much are you thorough with? Ask yourselves. Are you being disciplined? Are you studying every day? Are you practicing questions every day? Analyze yourself and start doing better. All right? I'll see you tomorrow. Bye-bye.